A living cell is also like a factory, or more like a group of factories, the cellular compartments or organelles. And if we could step inside one of these, we should find a number of cellular production lines, the metabolic pathways. Each step need not be physically linked to the next, but substances are broken down or synthesized in stepwise sequence in these pathways. And the cellular machines that speed up, catalyze each step are very specific in their activity. They're the enzymes. Enzymes outperform by far the catalysts prepared by chemists.
A single molecule of an enzyme is far too small to be seen, but you could picture it as a globular arrangement of thousands of atoms with a specially shaped place on part of its surface. That shape exactly matches the molecules of one particular substance, the material the enzyme is designed to work on. This enzyme breaks sugar molecules in half. As the sugar drifts past, it's drawn into place by the attraction between the atoms. And the forces pulling the sugar into the enzyme are so strong that they actually overcome the forces holding the sugar together, so it breaks apart. But as the atoms in the two broken halves rearrange themselves, they no longer fit the enzyme's shape, so they come away again. And molecules can be joined together in exactly the same way, but in reverse. It all happens very fast, although no other enzyme is quite as fast as catalase in the liver. Molecules of hydrogen peroxide are molecules of water with another oxygen atom attached. Catalase enzyme breaks off that extra oxygen and releases it as bubbles of gas.
Well, the first thing to understand is that enzymes are carrying out chemical reactions. They're not doing anything which is magic or mysterious or unique to life. What they are carrying out is very specific and very directed sorts of chemical reactions. Enzymes are a particular sort of molecule which performs a reaction known in chemistry as catalysis. That is, it's not used up, the enzyme, during the course of the reaction, but it makes possible a particular and very precise set of operations. Now, the most important thing about them, therefore, is that what enzymes can do is can direct a chemical reaction. Instead, for example, of a rather grand burning of sugar to give you carbon dioxide and water out at the end of it, the enzyme can make possible taking a sugar molecule and cutting it up and tailoring it into very specific other molecules as a result. So enzymes direct chemical reactions in a particular sort of way and produce rather defined end products which are useful so far as the cell is concerned. The other thing which enzymes make possible is reactions which might otherwise require rather special concentrations of acid or alkali, for example, high pressures or high temperatures. They enable the reactions which could only occur otherwise under these special circumstances to take place quietly and without much fuss in the living organism, in the cell itself.
So enzymes work as catalysts, directing chemical reactions and enabling reactions. They're proteins that perform very specific roles within living organisms. Enzymes control metabolism by allowing reactions to take place that would otherwise require great changes in temperature, pH level or pressure, changes that would kill the cell. Now, enzymes are a subject that come up a lot in exams. So look back over your notes, organise your thoughts, and then have a go at this exam-style question. How do enzymes interact with the molecules they work on? Have a go at answering the question yourself before carrying on.
One analogy could be a lock and a key. The way enzymes are configured is that they have a gorge or some kind of cleft in which the molecule that's going to be acted upon, that's going to be changed, that's called a substrate, falls into. So if you like, the lock is the enzyme and the key is the substrate. And what happens with any lock and key, when these form a complex, then the enzyme can go to work on changing the nature of the substrate. The induced fit hypothesis, as its name suggests, allows for the enzyme to actually perfect its role even further, And once it has formed a complex with the substrate, once the molecule that's to be acted upon has tumbled into the cleft, then the enzyme slightly reconfigures itself so that it's a perfect fit, not just any old fit, but a perfect fit.
You'll need to be able to go into this in greater detail. Make sure you can define activation energy and explain how enzymes work by referring to the active site, the enzyme substrate complex, the lock and key hypothesis, and the induced fit hypothesis.